The overall goal of the following experiment is to identify mitochondrial toxicity or beneficial effects of drugs using the model organism C. elegans. This is achieved by the in vivo quantification of ATP levels in transgenic C. elegans strains that express the firefly luciferase enzyme fused to the green fluorescent protein. The fusion gene confers two distinct properties to nematodes. The first is that when given the substrate luciferin, the nematodes emit light in the visible range, in proportion to their cellular ATP reserves. And the second is that when illuminated with the appropriate wavelength, they will exhibit strong green fluorescence. Fluorescence is independent of the nematodes' ATP levels and is proportional to their mass or numbers. Therefore, it can be used to normalize the bioluminescence measurements. During a typical assay, a population of developmentally synchronized worms is grown to the L4 larval stage. The nematodes are then treated with the test drug compounds for a period of time prior to the measurement of both fluorescence and bioluminescence. Results show potential drug toxicity when bioluminescence is reduced. An enhanced signal indicates a hit for further testing for beneficial effects on mitochondrial function.
The main advantage of this technique over existing methods, like drug screening on cultured cells, is that screening is being carried out in the physiological context of a whole live multicellular organism with close to 50% genetic homology to humans. And like humans, C. elegans actually requires mitochondrial oxidative